Today, we'll be discussing on the lytic cycle of viruses. So, as we all know from the previous classes that the viruses are the obligate intracellular parasites. So, one of the group of viruses that is known as bacteriophage, it infects the bacteria. So, they use the bacterial cell machinery system for its own multiplication and development. Bacteriophage, like all other viruses, they also contain the nucleic acid and proteins, and the genetic material may be either DNA or RNA, but never both. So the nucleic acid of the phages, they often contain unusual or modified bases which protect the phage nucleic acid from the nucleases that break down the phage nucleic acid during phage infection. Some of the examples of bacteriophages are the 5X174, T-even phages such as the T2, T4 and T6 that infects the E. coli, and temperate phages such as lambda and mu. So now we'll see the structure of bacteriophage. So bacteriophages, they have a different shape and sizes. In case of T4phage, it is one of the largest among the bacteriophages, and it is approximately 200 nanometer long and 80 to 100 nanometer wide. But generally, all phages, it range in size from 24 to 200 nanometer in length. So some of the phages may have the icosahedral capsid while others are filamentous. And inside the capsid, it contains a nucleic acid, and a capsid, it acts as the protective covering of the nucleic acid. And a hollow tube-like structure, which is known as tails, is also attached to the capsid, and these helps in the injecting their genetic material inside the host during infection. In case of T4phage, the tail is surrounded by a contractile sheet and a base plate like structure which is present at the end of the tail, from which the tail fibers are attached. And the function of the tail fibers is that it helps in attachment of the phage to the bacteria, while the contractile sheet, it helps in the contraction during infection. So some of the phages that do not contain these tail fibers at the end. Now we'll come to the general characteristics of bacteriophage. There are two main types of bacterial viruses, that includes the lytic or the virulent phage and the temperate or the avirulent phage. So during infection by the lytic or the virulent phage, the cells, it responds by producing a large number of new viruses, and at the end of the incubation period, the host cell, it bursts and release new phages to infect other host cells, and hence it is termed as the lytic cycle. Whereas in the temperate phase infection, the viral nucleic acid, it is carried and replicated in the host bacterial cells from one generation to the other without the involvement of cell lysis. Now we'll come to the infection of host cells. The various steps that are involved in infection includes adsorption, then penetration, then the synthesis of phage nucleic acid and proteins, assembly, and finally release. Now first we'll see adsorption. So bacteriophage, they attach to the specific receptor site on the host cell membrane, and the nature of the receptor, it varies with the phage. The cell wall polysaccharides and proteins, teichoic acid, flagella and pili, it serve as the receptor. In the case of T-even phages, the attachment begins when the tail fibers, it comes in contact with the appropriate receptor site, and the base plate, it settles down on the surface. This binding is carried out by the electrostatic interactions and is influenced by pH and presence of ions such as the magnesium ions and the calcium ions. So conformational changes it occurs in the base plate and the sheet when the base plate is seated firmly on the cell membrane and the tail sheet contracts. Further, the central tube or the core is pushed through the bacterial cell wall. So now we'll see penetration. The penetration of the phage into the host cell is mostly mechanical, but it may be facilitated by localized digestion of certain cell surfaces structure either by phage enzyme that is carried on the tail of the phage or by viral activation of the host degradative enzymes. So penetration in T-even phages is achieved when the tail fibers of the virus it attach to the cell and it holds the tail firmly against the cell wall, and when the sheet contracts driving the tail core into the cell through the cell wall and membrane, and when the virus injects its DNA. So the protein coat that forms the phage head and the tail structure of the virus remains outside the cell. So now we'll see the synthesis of phage nucleic acid and proteins. So after the phage DNA is injected, the synthesis of the host DNA, RNA and protein is halted and the cell is forced to make the viral constituents. The RNA polymerase, it starts synthesizing the phage mRNA, that is the messenger RNA. These mRNA and all the other mRNA, that is mRNA that are transcribed before phage DNA is made, it directs the synthesis of the protein factors and enzymes which is required to take over the host cell and it manufactures the viral nucleic acids. Then the host DNA are degraded to nucleotides by some virus-specific enzymes, thereby halting the host gene expression and providing the raw material for virus DNA synthesis. The bacterial RNA polymerase is modified by the other early proteins and a phage head proteins that are injected along with the phage DNA. Then the polymerase recognize the promoters on the viral DNA rather than the bacterial promoter sequence so that the virus genes are transcribed. Ultimately, within short period of time, the virus DNA synthesis commences. Then for the synthesis of T4 DNA, considerable preparation is required as it contains hydroxymethyl-cytosine instead of cytosine. This hydroxymethyl-cytosine, it must be synthesized by two phage encoded enzymes before the DNA replication can begin. So after this T4 DNA has been synthesized, it is glucosylated by the addition of glucose to the hydroxymethyl-cytosine residues. Then this glucosylated HMC residues protects the T4 DNA from attack by the restriction enzymes which would cleave the viral DNA at a specific points and destroy it. In T4 DNA, a base sequence is repeated at both ends of the DNA molecule, which is known as terminal redundancy. So after many DNA copies are made, about 6 to 10 copies are joined by their terminally redundant ends with the aid of several enzymes. So this long DNA strand, it is composed of several units which is linked together with the same orientation known as concatenamers. During phage assembly, these concatenamers are cleaved in a way that the genome is slightly longer than the T4 gene set. Now we'll see assembly. So the assembly in the T4 phage is complex. The lead messenger RNA directs the synthesis of three kinds of proteins. That is, the phage structural proteins, then the proteins that helps with phage assembly without becoming part of the virion structure, and the proteins that are involved in the cell lysis and phage release. In T4 phage, the lead mRNA transcription begins about 9 minutes after the DNA is injected into the bacteria, and simultaneously all the proteins that are required for the phage assembly are synthesized. After the base plate is constructed, the tail tube is built on it and the sheath is assembled around the tube. Then the procapsid of the phage is constructed separately of more than 10 proteins and then combines with the tail assembly. Then the procapsid is assembled with the help of a protein which is known as scaffolding proteins. Further, the tail fibers are attached to the base plate after the head and the tail have come together. Finally, the DNA is drawn into the complete shell efficiently. Now the final phase is the release. So many T4 genes are involved in the release process. One directs the synthesis of a lysozyme that acts on the peptidoglycan layer of the bacterial cell wall, and another phage protein, it damages the bacterial plasma membrane and allows the lysozyme to reach the cell wall beyond it, and it inhibits the energy metabolism reaction that is located in the membrane. Now we'll come to the multiplication cycle of bacteriophage. So the sequence of events that is initiated by the injection of the phage nucleic acid and culminating in the release of newly synthesized virion is termed as the viral multiplication or the replication cycle. So this viral multiplication involves two phases of period. The first phase is the eclipse period and the second one is the intracellular accumulation phase. So first we'll come to the eclipse period. During the eclipse phase, no infectious phage particle can be found either inside or outside the bacterial cell. Then the phage nucleic acid, it takes over the host biosynthetic machinery and the phage specific mRNAs and the proteins are made. So there is an orderly expression of the phage directed macromolecular synthesis just as one sees in the animal virus infection. Early mRNAs, that is early messenger RNAs, it codes for the early proteins which are needed for the phage DNA synthesis and for shutting off host DNA, RNA and the protein biosynthesis. And in some cases, the early proteins actually degrades the host chromosome. After this phage DNA is met, late mRNAs and the proteins are also met. The late proteins are the structural proteins that comprise the phage as well as the proteins that are needed for the lysis of the bacterial cell. So now we'll see the intracellular accumulation phase. At the end of the eclipse period, mature phages, it begins to accumulate intracellularly until they are released by the cell lysis. In this phase, the nucleic acid and the structural proteins that have been made are assembled and the infectious phage particle, it accumulates within the cell. So no newly released extracellular phages can be seen until the lysis begins. Hence, the time interval from the infection until lysis is termed as the latent period. And at the end of the multiplication cycle, the phage number, it continues to increase until it reaches the constant titer, and this time interval is termed as the rise period. So that was about the lytic cycle of viruses, and I will wind up by adding that lytic cycle of the virulent bacteriophage is a life cycle that ends with the host cell lysis and the virion release. And the life cycle of the T4 phage is composed of several phases. In the adsorption phase, the phage is attached to the specific receptor sites, then followed by the penetration of the cell wall and the insertion of the viral nucleic acid into the cell. Finally, the complete virions are assembled immediately after the separate components have been constructed and the phages are released upon lysis.